It's the first time I've had to share slides with Zoom. We don't use Zoom. Sorry, everyone. I need to quit and rejoin. I apologize to allow permissions. My fault.
Yeah, no problem. Yeah, and I have to wait and let Dr. Fulton in again. Just a second, everyone. There he is. And there we go. Sorry. Oh. Let's see. Ben, can you hear us? We see the slides, but I don't.
Can you hear me now?
Yeah, we can hear you now. Thank you.
All right, perfectly.
Yeah,
I was saying that I am completely, unlike everyone else in the world, I am completely unfamiliar with Zoom. We don't use it at Regeneron. So it's the first time I've actually used it. I had to get special authorization from our IT department to install it today. So I apologize. Anyway, so as I've been introduced, I'm a scientist in the infectious disease department at Regeneron, and generally I have a lot of training in RNA viruses. I just joined the company 2.5 years ago, and straight from grad school where I worked on measles, Zika, and flu virus. Since then, I've been working on obviously SARS-CoV-2, but also Ebola virus. And I was asked to just give an update basically on the COVID cocktail that we've generated, try to explain how it works and how it's different than a vaccine. But I also wanted to take the opportunity to basically also introduce who Regeneron, or who we are at Regeneron, what we do and why we're kind of a special company and what's special about the COVID cocktail that we also helped develop. So with that said, I would like to hear your questions as they come, particularly if you're confused about something or you have something that you might be pertinent and you want me to go into more detail. I'd like to keep this more as a conversation, if that's all right with everyone. All right, so starting very basically, we're very close by to Roxbury. We're just in Westchester in Tarrytown, where our main campus is. So we're very close by. I actually live down here in Manhattan, and reverse commute, where I used to reverse commute before everything was closed down. So I also want to say we're a large biotech company, but compared to a lot of the other companies that have been jumping into the COVID space, we're actually pretty small. We're not a big pharma company. We're very much a biotech company. And that allows us to be a little bit more focused and a little bit more science driven, which is a great benefit. So just jumping ahead. So this is our campus. Where I used to work. I don't work there I haven't been there in 6 months. And this is my building right here. This is where all the research is pretty much done for Regeneron. We also have facilities up in Ireland for production of our product in the EU and Europe. But we also have a production facility in the Capital District, in Rensselaer, New York, where we actually produce all the drugs. This is just where we do all of our research. This is a very important site for us. Well, this is a pretty big campus. This is very small relative to a big pharma company. We are Very unique that we do all of our research, and we actually have a tremendous number of researchers compared to our total population that works here. So the percentage of our research team versus those involved in like legal or management and other things is very high. Okay. Despite being a relatively small company in terms of the pharmaceutical industry, we actually have a tremendous number of products already approved. We have 9 FDA approved medicines. I'll get to why this is actually a really staggering number based off of our size of our company. But I think what is most unique about us is that basically every drug we make has been researched and designed in-house at Regeneron. And this is not something that's usually done, particularly in companies that make a lot of drugs. Usually a lot of companies buy basically ideas that have already been formulated, drugs that are already in development, and then just finish up the manufacturing process to get it into the clinic. But we really develop everything in-house from the very start of the project to the very end. And that's something that is very unique. And we're not happy just sitting on the 9 FDA approved medicines we have. We have at least 30, this probably numbers changes very frequently, that are in clinical development. So these are drugs that have already gone through the whole process, from the clinical development, the basic research to the clinic, and hopefully these will be approved. But what I really, really want to stress about us, and I think it's very important, and which makes us unique, is that we do a lot of basic research. The primary job of what I do isn't like clinical research. It's not just trying to get something in the clinic. It's also just trying to understand the mechanisms. What is the best way of developing a drug? And I think that's reflected in the number of peer-reviewed publications that we have in terms of each year. And, we've had up to 175 in 2020. And this is a really impressive number, and we're happy to share our science with people. Let's see. Oops. So I know that next week there's going to be another talk on drug development, but what I want to jump ahead of in terms of also just describing how impressive this year has been, not just for Regeneron, but also for science in general, is the speed at which we develop drugs. And I'm not just talking about Regeneron again, I'm talking about the global response. It is truly staggering. So drug development is a very time-consuming and very expensive process. So most drugs take at least 10 to 20 years to develop on average. And in terms of when you get through the clinical development, it's going to be $1 billion or so plus usually in terms of the investment into that drug. Unfortunately, even if you get it to the clinic, which is that time where you've already sunk all this money into it, very few percent actually make it through in terms of approval. It's less than 8%. This fluctuates year to year, but this is around what it is. And a really, really important thing, and I think an area where we have a lot of, in terms of room to grow, in terms of just a society in general, and also in terms of pharmaceutical industries in general, there's only about 10 to 20. I mean, this can fluctuate a year. Some years, definitely, particularly in recent years, there's been a few more than this, but only 10 to 20 drugs are approved each year. And I think this shows there are more than 10 to 20 diseases we need to address each year. So this shows like how much room we have to go better as an industry or as a society in trying to improve this number. And it's actually what's really important is less of half of these are actually novel. Most of these drugs are reformulations or recombinations of drugs that have already been developed. So I just wanted to illustrate this, and particularly with the hindsight of like how much has been developed and how much has made it to the clinic, including the vaccines from Moderna, J&J, Pfizer, also the other companies that have developed antibody responses, the antibody cocktail therapies. This is very impressive for the past year. Okay. I don't want to go through that much detail about this, but I think what also makes Regeneron very unique is we have a very broad repertoire of diseases or that we're trying to target with our drugs. So as I said, we have 9 approved therapies. These target anything from cancer to macular degeneration to high blood pressure or high LDL. Also, we have two. We have a infectious disease one. We have a approved therapy for Ebola now. So we don't just focus on one area like a lot of companies do. We really spread out. We go with what we're interested in, what we find, would be the best benefit to the society. It's also reflective in terms of what we actually have in clinical development right now as well. We have compounds that are being generated for basically a bunch of things. We have a lot of oncology, which is the orange pockets. We have a lot, again, in inflammation and immunology as well in development, infectious diseases. But not all these things are, probably going to be big sellers. We're really interested in the science and trying to help people. One example of this is this activin A targeted drug. This is for a very rare disease that affects, I think, less than 1,000 people globally, but we still want to get it out there to help the science and help these people. I think that actually is like a pretty good hallmark of what makes us very unique as a company. So there's a lot, and this is for Regeneron, there's a lot that goes into developing a drugs like the COVID cocktail. A lot of different departments, from the initial, target discovery, like what disease do you want to target, the mechanisms of which you can induce to get there as fast as possible, like what facilities and departments you have that are ready to work on product and get things through our pipeline, which is basically our drug development process, into the clinic. And this goes from just the tech dev to the manufacturings, to the medicines, to the scaling up, to actually getting it in enough material to go in the clinic. And this is actually a major a feat that is very important in terms of this drug discovery, particularly for these monoclonal antibody therapies. Where I sit in this is this tech dev. So basically, we take this target and we just try to develop technologies basically to help us get that technology or that target that we want into the clinic as fast as possible. And so what I got hired to do was not at all to do with infectious diseases, or not traditionally involved in infectious diseases. What I got hired to do at Regeneron was to develop oncolytic viruses. These are viruses that we engineer to basically infect tumor cells and infect and kill them, or basically induce a very strong immune cell to that. But it requires a lot of viral engineering, which is perfect, and why I got basically recruited to the SARS-CoV-2 team when this outbreak occurred from the very beginning. Just because I had this molecular and viral engineering skill that allowed us to develop the tools to basically test our antibody therapies against the virus. Another thing that makes Regeneron very unique is that we have this velocity suite pipeline, basically. Again, this is just our ability to start with an idea and get that drug through the pipeline of all this development into the clinic. And I think the two most important ones that we have for what we did for SARS-CoV-2 were the VelociMune and the VelociMab pathway suites that we have. So the VelociMune is basically, I'll get into this in a little bit more detail later, is basically we've engineered mice to have human antibody responses to antigens, or as close as we can get to them. And so that we can take these humanized antibodies from these mice, instead of having to vaccinate humans to get the antibodies, we can vaccinate these mice. And then we can take those antibodies and then develop them into the therapies. If you use a traditional mouse antibody in a human, it's going to be immunogenic, which means your body is going to recognize it as foreign, and it's not going to be accepted very well. So if we use these humanized mice for the antibody responses, we can take these mice and supply these antibodies to humans without developing this immune response. And they're basically, in all purposes, human antibodies. We also have this VelociMab suite, is basically the ability of go from this single antibody that we want to develop and get it from this very low scale, like very small scale amount to getting it into these very large bioreactors for producing large amounts of these antibodies very quickly. And I can talk about that more in a little bit. So I know last week, Mark Jenkins gave a really good overview of antibody responses. I actually watched his lecture and I thought it was really, really on point and very, very good. So I'm not going to go into too much detail, but just as a reminder, antibodies are proteins, just like what you would find in meat or what other food you eat. And they are very special, and they're produced from a cell called a B cell. And this B cell basically can undergo mutations that make in the antibody genes that allow them to make regions of these antibodies highly variable. And so each B cell will make a different antibody that can be variable in this region that is used to bind antigens, such as those of SARS-CoV-2 or other virus particles or bacteria. And so because of this, you can get very highly specific responses from individual B cells and antibodies to whatever antigen you want to target. These B cells, that produce these in terms of like when you actually get vaccinated or infected, they can basically remember this antibody because they have it encoded into them, and that gives you long-term immunity to those pathogens because they have these B cells that express this antibody and can basically continue to pump it out for hopefully the course of your life and keep you immune to whatever you've been exposed to. And why are we interested in antibodies? So this is the major drug that we make at Regeneron. We make some other drugs too, but the primary one are these monoclonal antibodies. And the reason why we're interested in them is they make really good drugs in a lot of ways. And when you think of drugs, you're usually thinking of something like a small chemical, like ibuprofen, antibiotics, aspirin. Antibodies are much, much bigger than that. These are real proteins of, much larger than, say, aspirin. And this has a couple of different advantages. So small molecules, and there's some really, really good small molecule drugs out there. I'm not trying to say they're not all good. It's just that when they're small, they can have a lot of off-target effects. It's really hard sometimes to understand what they're doing when you give them to a patient. With a monoclonal antibody, they can be very, very specific to binding that protein that you've generated against them, and they'll have a very specific function. The other thing that makes them advantageous is if you give drugs such as antibiotics, often you have to give so much you have to give a high dose of this to build it up in your bloodstream or wherever you need it, that it's actually almost to the point where it's becoming toxic. And these drugs are actually cleared from your body very frequently, so at a high rate. So if you give it, it might be gone by the end of the day if you give it in the morning. This means that you have to give constant doses, a couple times a day often, as you all experienced with probably getting antibodies or not antibiotic treatments in the past. The reason why you have to give it multiple times a day is because you don't want to get to a toxic level, but you need to maintain that level in your body. To just maintain whatever function it's going to do against the bacteria. But with a monoclonal antibody, these things tend to not be so toxic, particularly if they're targeting a pathogen. You can give high doses of these, and they actually persist a very long time in your body. Since these are large proteins, they can circulate in your bloodstream for up to a couple of months. And this means that you can give them, and you can give one dose of them, and you should be protected for a while. And that's like one advantage, theoretical advantage for these drugs compared to a small molecule. I also wanted to talk about why they are different than vaccines. I think Mark Jenkins gave a really good overview of this last week. But vaccines are really different from what we're doing in a lot of ways. I mean, in a lot of ways, they're also pretty similar. So vaccines, you're basically supplying an antigen, so a virus or part of a virus or a bacteria, and you're injecting it basically into your muscle or wherever it needs to be injected for the activity of that vaccine. And your body is recognizing it as foreign, developing antibodies to that produced from these B cells. And this has a lot of advantages because one, they're really good at developing long-term protection. These B cells that you produce to these antigens can exist for a long time, produce a lot of these antibodies, and basically give you almost lifelong immunity to a lot of different viruses, particularly something like measles. What we're doing is something different. We're basically trying to capture a couple of these antibodies from, like, would be produced basically by a vaccination. That would be the best for doing whatever you want, such as protecting you against a virus infection. So we're trying to get the most potent antibodies from there. And basically what we do is we take them out and we clone them separately from B cells that are expressing them. And then we produce them basically in tissue culture in a petri dish or a large bioreactor, which is like a large fermenter basically. And then we collect the antibodies from those cells, and we then can purify them and then supply them to patients who have not seen the virus before. And this has a couple of different advantages, because vaccines, when you supply them, you need to give time for you to develop an antibody response. And this is somewhere around a couple weeks to, as you know, with Moderna or Pfizer, to get the full protection, you need to go 2 weeks from your second dose. If you supplied this during an acute infection like SARS-CoV-2, it's not going to do much for you because you already are developing antibodies to the SARS-CoV-2 pathogen. So this is not going to be great for treatment. It's really good for prevention, though, long-term prevention. What we're doing is, since we're already supplying antibodies that are fully developed, highly neutralizing or potent against the virus, you can supply it to a patient that is acutely infected with an infection and actually suppress those viral titers and develop protection that way. But this is going to be short term. This is only going to be dependent on how long that antibody can actually circulate through your bloodstream over time, since you haven't developed antibodies from your B cells. So there's no B cells in you expressing these antibodies, producing them. So this is very temporary. This is temporary, but it's very effective. And so you can use these both for treatment and prevention this way. At this point, are there any questions? All right, I'm going to continue on.
So I don't see any in the chat yet. So.
All right, great. That's good. So we start this process by cloning out individual B cells that are expressing the antibodies from vaccinated mice or people. We take these B cells out and we clone out the antibodies from them and start producing them at small scale. And this allows us to screen these antibodies for the effect that we want. And we usually screen thousands of antibodies for whatever effect we want. And then we narrow it down from there. Once we have a few that are of interest, we basically go into, if anyone's a home brewer or anything like that, I used to be a home brewer, we go into like a homebrew scale, like 5-gallon production facilities for our leads of antibodies that we're interested in. And this is all done on Terrytown, the campus that I work at. And this allows us to get into relatively large scale production of these antibodies that we can continue working with them. And then once we have clinical candidates, these go up to Rensselaer and the Capital District. And these go into really large fermenters like you would see in a professional brewery, actually way more advanced, but very similar in terms of the scale. This is done underneath extraordinarily careful conditions in terms of production, where we have to ensure everything from the quality of the cells to the quality of the antibody. It's very time-consuming and incredibly difficult process to do. And what we've developed over time is the ability to go from this bench scale to a large scale very rapidly, much faster than I think anyone else in the industry. And it makes Regeneron very unique. And this actually allows us to respond to outbreaks or pandemics or epidemics much more rapidly than other people. Not only do we have this suite of technologies that are upstream in terms of the drug development that we have from the Velocity mouse suite that we have, we also have this ability to get whatever drugs or antibodies we want to develop into these reactors very quickly and develop large-scale clinical batches of these. And for this reason, we've actually started several rapid response programs for pathogens that have emerged. Prior to SARS-CoV-2, we had Ebola and MERS that we started developing for. And our timelines back then were unprecedented. We went from basically idea of the isolation basically for Ebola virus in 2015 to isolating antibodies, testing them, and then taking our leads within 6 months. We then were able to then, once we got them into our manufacturing step, we were able to go through and getting them into the clinic within another 6 months. Got a little bit better with this the second time with MERS. We got it down to like 3 months, I would say 9 months total. But this helped us, since of all this experience we had here in terms of developing these other drugs. When SARS-CoV-2 happened, we were really prepared to actually start the process and get these antibodies into the clinic as fast as possible. And I think we did it in less than 6 months from basically concept to the clinic, which is just amazingly unprecedented. If you think about it, generally it takes most drugs 10 to 20 years to be developed.
Dr. Fulton, I do have a couple of questions.
Yeah.
If I am not interrupting you.
No, absolutely.
Are you working on the variant booster?
The variants?
The variant, oh yeah, sorry, the variant booster.
Yeah. The variants, I generally, my job right now entails basically generating all these variants, testing them, and surveying what is out there right now, and making sure that our cocktails are still effective against them. And so far, it looks like everything that we've seen, the cocktail is still effective.
Another question, will the short-term treatment of the monoclonal AB interfere with long-term immunity process?
That's a very, very good question. I think in people who are uninfected, it shouldn't. But if you have a cocktail therapy and then you're getting vaccine, I think it's something that needs to be tested. But I can't really answer that question right now, but it's a very, very good question.
Thank you. One more. What are the criteria for a patient to be accepted for MCA therapy?
I should have said this from the beginning. I am not a clinician. I'm very much a basic researcher. Honestly, my family will call me with updates from the clinic in terms of our approvals before I even know internally. So I'm really not qualified to say that at the moment. I can refer you to the website, which will have you all the information you want for that. I don't want to say anything wrong. But our first approval was for mild to moderate COVID in high-risk patients for the EUA. I think it might have been a little bit broadened out from that since then.
And one more. Where does growth media go after harvesting? Is it cleaned up before discharge?
Oh, I actually don't know. I'm sure since it's probably going to be considered a biohazard, it's probably going to be at least autoclaved. So basically heated at very high temperature to destroy the biological compounds in that. But I don't know. Sorry.
All right, those are the questions I have for now.
Those are all really good questions. Keep them coming.
That's right.
All right. So a little bit more on this. So we knew we wanted to make, as soon as the outbreak occurred, we knew we wanted to start making therapies for this virus immediately. It was clear from all the data that was coming from China that this was going to be a problem that was going to hit the United States. Also, even if it didn't, it was still going to be a big problem in Asia, and that we wanted to help solve this, this epidemic. So the way antibodies for this virus work is that SARS-CoV-2 and other viruses, they are obligate intercellular pathogens. So in other words, they need to replicate within cells. They can't replicate by themselves without cells. So if you can block their entry into cells, you can prevent them from replicating and you can stop the infection. And so the way these monoclonals work is that they block the interaction of the spike on the surface of SARS-CoV from interacting with ACE2, the receptor for this virus, and therefore block entry. And we knew from our past experience with other pathogens that we wanted to have at least two antibodies targeting the spike at the same time. And I'll get into why that is really, really important now. I also want to talk about variants as well, since it's already been brought up. And it's something, viral evolution is something that I have thought about a lot. You know, it's like what I did my grad school research on, and it's something I continuously think about all the time. So viruses, and particularly RNA viruses like SARS-CoV-2, can actually mutate very quickly and develop a lot of diversity. And what I mean by that is a single virus, if it infects a cell, as it's copying its genome, it can be very bad at doing this. And it will induce genomic typos basically into the genome. And this will potentially create those variants that we've been hearing about in the news all the time. And so a single virus infecting a cell can develop a fair amount of diversity. Most of these mutations, as you might imagine, actually are bad for the virus, and these genomes will be lost. Because if you have a mutation in a critical site that's required, for example, receptor binding, that variant can continue on and it will not be a viable variant. But a lot of these mutations are neutral or sometimes can actually be beneficial for the virus. And so when this virus buds out, you can get a pretty diverse pool of variants in your population. And this is actually really important in terms of how these viruses evolve. And a lot of the news that we've been listening to recently is that they can develop a tremendous amount of diversity. And the other thing that's really important is a single virus, maybe this is not true for SARS-CoV-2, but other viruses that can produce thousands of progeny from a single virus. So one cell getting in can make thousands of viruses. Another thing that's really important is that these populations of viruses are huge. I mean, you can have some viruses that a single teaspoon or a drop of liquid can hold 1 billion viruses. Particularly combined with the diversity that these viruses can develop, you can get a huge amount of diversity and a large population here. That means if you put selection on these viruses, you can start selecting for variants that have greater fitness than their counterparts. And one of the things that is often, selective pressures that's often put on viruses comes from antibodies. And this comes from both like our natural immune system, but this can also come from antibody therapies that people have developed. So let's just say that you have an infected individual. They have a large population of diverse viruses in them due to this mutation rate and this high population load that's in them. You add an antibody to them with this population to try to block or prevent the infection. What can happen is you'll neutralize most of the viruses, but you might have a virus that can survive. And this could be due to the fact that it can encode a mutation that actually prevents antibody binding and therefore neutralization. And after multiple rounds of replication, you can actually get a virus that can then continue on and develop a drug-resistant population. And so this is like a worst-case scenario when you're developing a drug. This is not what you want to have happen, obviously. So you want to try to prevent this as much as possible. And this is broadly, not like identically, but can be also what happens with bacteria and bacterial resistance that we've been hearing about for years. It's not exactly the same mechanism, but it's similar. It's not that you're basically the antibody binding to the virus is forcing the evolution of a new variant. It's just that by selecting with an antibody that can neutralize most of the population, you can select for a variant that already exists in your population. I think this is important to understand in terms of how these viruses evolve.
I have another question if...
Yeah, sure. I figured I would have with this.
Does the anti-spike response come up before the response against core or envelope? Why didn't go after core proteins or envelope proteins, which don't mutate?
Great question. So we want something that's on the surface of the virus so that we can target it and prevent entry. The other thing is you need to block something that is absolutely critical for entry. So the easiest one to go after is receptor binding, which is encoded for by the spike. And so that's the number one reason why we did this. I'm not saying you couldn't develop drugs to them, but this, in terms of like practicality, it's the easiest way of blocking that initial round of infection and preventing you from even developing that adversity in the same first place. Did that answer the question?
Yeah, thank you. That's it.
Okay. So we really knew we didn't want to have this situation. And we did a lot of experiments, and we realized that we really needed to have at least 2 antibodies in our cocktail to prevent this rapid mutational escape. So in the same situation, if you take your large population of viruses, you add in your antibodies, this time 2 different antibodies, such as what we have in Regeneron cocktail. This time you're more likely to neutralize a greater percentage of your population. And this is one being that if you have 2 antibodies binding distinct regions, if you have a variant that can block neutralization by the one antibody, you're unlikely to block neutralization by the other. And this is very, very critical. They must bind different regions of the spike. And we've shown this experimentally in a lot of our escape data. We've published several papers, some in Science recently, looking at this. If you don't have them binding distinct epitopes, this mutation can affect both antibodies at the same time. Epitopes are just the target sequence for the antibody. So it's very, very important to have them binding distinct regions. So this is the way that we decided to develop our drug. And we actually learned a lot of this from our experience with Ebola, which was our Ebola therapy, which was just approved by the FDA. So we knew we wanted to do this because this greatly, greatly diminishes the risk of drug resistance. Not only that, let's just say with all these variants that are coming out recently, it also diminishes the likelihood the variant would already have an escape variant that's going to be affecting your antibody neutralization. By having 2, the variant would have to have 2 mutations in them at the right spot to block your therapy. And so far, we haven't seen those appear. All right, so to develop these 2 drugs, we actually got our antibodies from 2 different sources. One from our Velocity mouse, which is the mice that have the humanized antibody responses. And we also got one of our antibodies from a human patient who recovered from COVID, basically. And we collected B cells from this. We then cloned out the antibodies from these B cells. We just basically isolated all the B cells individually. And we basically screened around 3,000 B cells, antibodies from B cells, for the ability to bind and SARS-CoV-2 spike. From that, we then tested neutralization against the spike, preventing infection. We ended up with 48 that looked promising. But since we wanted to have this non-competitive or these antibodies that are binding 2 different regions of the spike, we then pared this down to a smaller segment of 9 antibodies, and then finally down to 4 and 2. So this is what we ended up with in our clinical product. And I can't stress how amazing this timelines are. I remember getting a call from my manager in January. I actually had the flu at the time. She asked, how quickly can we develop this, start making the immunogen? So that's what my group, I lead a small group of scientists that do the work in the lab. And they're like, how quickly can we develop an immunogen for this virus? And I'm like, we can do it in a month. They're like, no, this is really important. We need to do this now. And it was very clear at the time that we needed to jump on this. And we were able to clone this immunogen immediately. And so the majority of this time that we have in terms of this vaccination was actually just waiting for the humans and the mice to actually develop antibody responses so we could collect them. After that, it was more like that was the first 40 days basically of this. The last 30 days were all this production and downstream screening, which happened in crazy timelines. Really, really fast paced, very impressive how the company just really came together, recognizing the risk and what we needed to do to get this done as fast as possible. It was an amazing time to be at the company. Since then, since we have these 2 clinical products, we got them into the clinic. There are several ongoing clinical data. And actually, this is a slide from late last year. So this is actually out of date. Some of these clinical studies have some data already where I'm going to go through some of the data in a minute. But they're not, these clinical studies are not over. We're continuing on with them. We want to really make sure that these are the best drugs possible and understand how they're working in the patient population as well as possible. But what we've learned so far, the way we got the emergency use authorization was that we saw in a study that compared to the placebo, we were able to reduce viral load, particularly in at-risk patients. This might not look that impressive, but this is a log scale, a 10 graph. So this is a pretty big difference in terms of viral load. And it also greatly reduces the medical visits for at-risk patients compared to our placebo. And so this is the data that got us the EUA. Since then, we've found clinical data that shows that we really reduce hospitalization death by 70%, which is amazing. We also looked at basically households where one patient got infected in the surrounding household. We looked to see if we could prevent infection in the surrounding household. And we realized we could actually get really good protection for the surrounding households, even after they were already infected asymptomatically, and they would not convert to symptomatic infection. We also saw a really good protection in terms of people who had really severe disease already, reducing the risk of death or mechanical ventilation. So I think, basically starting on this drug over a year ago, just a little bit over a year ago, this is very impressive to actually get to this point where we have this data. And these numbers are really impressive in terms of what we have. And what I actually want to stress here that I think would have been really helpful, and also just like a little plug, is I think testing is absolutely critical for doing these studies. This is an acute infection, so I think we could actually improve this numbers by having greater testing. The earlier on you can give this therapy in terms of when a patient is infected, the greater the outcome will be in terms of preventing infection. If you assume that basically if you give the drug after the person has peaked viral titers, it's really not going to do as much as you can give it if you give it early on and prevent you ever from reaching the peak viral titers. So I think we could actually do even better if we had a little bit better testing here as well.
I have a couple of questions, if I may interrupt.
Sure.
Dr. Fulton, what's the window of opportunity from a time perspective to give the MCA treatment following COVID-2 infection?
I don't actually know. And again, a good question. I can't answer that much about the clinical stuff. I'm sorry.
No. The other one is, how is it given? IV or IM?
IV generally. For this
Okay.
asymptomatic, it was given, I think, IP. Or not IP, intradermally. I could be wrong though. I would have to look at that.
And can the MCA treatment be done outpatient, or must patients be in the hospital?
Oh, I think it can be out again. I'm going to defer this question. I don't know. As I said, like they keep us very separated from the clinical data. I'm really a basic scientist, and I honestly hear most of my clinical information as it hits the news from my family rather than from the company.
Understood.
Okay. So yes, we got the EUA, which was a really amazing day for us. It was like surreal. back in right before Thanksgiving. And we had already taken a huge risk at the company to basically start scaling up this production before we even knew it was 100% effective in patients. And it was an amazing, it was a great gamble by the company that it paid off very well. And this is the first treatment of any kind that really had a statistically proven effect on reducing viral load and outcome. So this was a very great day for us. As I said, the clinical studies are not over, and getting back to someone's questions earlier, we're not done. We're not complacent here in terms of, we've developed a drug, we can now walk away. We're constantly monitoring new variants that appear. My group has cloned over 100 and generated 100 of these variants already and tested them against our antibody therapy. And not a single variant has knocked out, as far as we can see, the combo by itself. We do sometimes see loss of one of antibody over time, depending on what mutation the variant has, but we have not seen loss of the cocktail. So this is really impressive, and we're continuing to screen other antibodies, basically, in case we need to replace one. Let's just say a variant becomes very popular, we can always replace it with one that's going to be effective against the variant that exists. So that would be the idea. So that's basically what I have. I just, as a concluding remarks, it's just a plug for the biotech industry in general. I mean, I think this year has shown, or this year and a half now has shown how much our society can be affected by these viruses and how important it is to put money and research and education into developing scientists who can actually help develop these drugs, because we don't want to be in this situation again. This has been costly, not only from a health and life point of view, but this has been very costly economically as well. I also, as I said from the beginning, I'm really impressed with how the society has done in terms of not just our own drugs, but also other companies as well, in terms of just being able to develop these therapies. And I hope we continue to take this kind of motivation and this streak to start applying this to other diseases as well. So that's it, and I'm happy to take any additional questions.
I have one. Have you considered going after reperfusion damage, probably, which is what is happening in the terminal phase of COVID?
Yeah. So that's definitely an intriguing question, because the virus induces a fair amount of damage, but your body, in terms of the response to the virus, also induces a fair amount of damage as well. And that's actually the feeling you get of sick is actually from the cytokine loads that you get, or interferon or other types of molecules that you produce in response to the infection make you feel crummy. So yes, we've definitely considered it. That's all I can really say about this, though.
Anyone else? Questions for this evening for Dr. Fulton? Please feel free to put them in the chat. Maybe I have a question. Is this the antibody cocktail? Is that for young and old? Is it, let's say my son had corona, he's 19. Would that be beneficial for him, or is it age? I mean,
yeah, I would have to defer again, saying I'm not a clinician.
Oh yeah, that's again a
clinical. But I would say that it depends on the situation, right? So I think this, so far, it has proven to be a safe therapy based on the data, and I think it's beneficial for people who are developing COVID. I think personally myself, I would take it if I was at risk.
Thank you. Well, Dr. Fulton, I don't see any other questions. Thank you so much for joining us. There's one coming up. I didn't understand the slide where you immunized mice with convalescent lymphocytes.
Oh, okay, yes. Sorry, let me clarify that. This slide, correct? We didn't immunize mice with convalescent humans. Basically, we had two approaches to making these antibodies. One was from our humanized mice. We vaccinated them with SARS-CoV-2 spike to develop antibodies, humanized antibodies to the spike. We then isolated these B cells and added them into our screen in terms of selecting B cells that were neutralizing. We also, for one of our other antibodies, we took B cells from convalescent humans, isolated them, and put them into our screen as well. I hope that clarifies that question.
Another one, any evidence that your therapy can be used to treat long COVID?
Ooh, I do not know. I'm sorry, I can't answer that question.
Oh. Let's see.
Yeah, it's disappointing. I apologize. I have to, be very careful of what I'm saying.
I have another one. If you have been vaccinated and become infected, would you be a candidate for the antibody?
Again, I can't say for sure, but I don't see any reason why not, particularly if you're infected and you're qPCR positive. I think it would be a very good reason to, it would probably imply that you have not developed a very good antibody response, and you would be a very good candidate probably for receiving this medicine. Because that's I have a feeling, yes, as vaccination rates go up, we're going to have more and more people immune, but there are people with breakthrough, infections. And I think that's really where this drug is going to shine in the future. People who do develop, are immune compromised and don't develop an immune response to begin with, or develop a suboptimal response. I think this is going to be really important for that population.
Great. Ron asks if Lynn, Westchester Wildcats win the tournament, did she score any goals?
No comment.
All right. Well, thank you everyone for joining us, and special thanks to Dr. Benjamin Fulton. Thank you very much for this very informative and interesting talk about the antibody therapy. Please join us next week, same time, Wednesday, May 12, about drug discovery and infectious disease by Dr. Cyril Kuhn, and he's from Boehringer Ingelheim. So another interesting speaker in this lecture series. Thank you, everyone. Have a good evening. Thank you for joining us. Thank you.
Thank you.